In this study, we sought to integrate artificial intelligence in genomics to identify alternative and novel mechanisms of inactivation of CDH1 in lobular carcinoma. As we know, lobular carcinoma represents a strong genotypic-phenotypic correlation in breast cancer, given that CDH1 biallelic mutations are pathognomonic for lobular carcinoma. There is a very high frequency of CDH1 biallelic mutations, like 80% of lobular carcinomas have them, whereas non-lobular tumors very rarely harbor these genetic alterations. So this very special molecular underpinning results in unique genetic vulnerabilities that are a tremendous opportunity that could be potentially exploited therapeutically. There are challenges, however, that hinder our ability to exploit the uniqueness of these tumors, such as, for instance, the fact that not all lobular carcinomas harbor CDH1 biallelic mutations. There is a subset that lack genetic alterations in CDH1, and the fact that the inter-observer agreement for lobular diagnosis based on histopathologic examination is low or modest. And that's why in this study we sought to come up with a genetic ground truth instead of a histologic ground truth. And we trained an AI model to predict the presence of CDH1 biallelic mutations without using histopathologic features or pathologic diagnosis, based on whole slide images. And then we applied this AI model, genomics-driven AI model, to study those tumors that were predicted to harbor CDH1 biallelic mutations, but according to our targeted frequency data, did not. And our study of these cases that we call the false positive cases through genetics and epigenetics resulted in the discovery of alternative mechanisms of inactivation of CDH1. Such, for example, we observed that a large proportion of these lobular carcinomas that lack CDH1 genetic alterations show CDH1 promoter methylation. So CDH1 promoter methylation is prevalent in CDH1 wild-type lobular carcinomas. And we, through the integration of AI and genomics, we also identify novel mechanisms of inactivation, such as a deleterious fusion affecting CDH1, associated with loss of heterozygosity of the wild-type allele, as well as intragenic deletions of CDH1 and non-coding genetic alterations affecting this gene in the form of pathogenic splice site variants associated with loss of heterozygosity of the wild-type allele. Taken together, this shows how the integration of AI and genomics is a robust approach to identify novel biology.